Welcome everybody to the December MAIC webinar. We are super excited to have you with us again this month. Before we get started with Tom Kiffney presenting, I just wanted to do a couple announcements from MAIC. So first, all of it is NACE, first of all. So NACE conference updates, everybody. So we have the NACE workshops that are starting to fill up. A couple are already full. But if you're interested, you should definitely check out the workshops and field trips on the website. They are really awesome this year. So definitely get registered for those as soon as you can. Also, if you haven't already, you should register for NACE because we'd love to see you there. And finally, Hotel Early Bird for NACE is ending next Friday, the 22nd. So you can still book after that, don't worry, but it does get more expensive, so you should do it before then. And then just as a note for next year, oh my gosh, that's crazy, 2024 January. On the 19th, we will be having our January webinar. It's again the third Friday of the month, and that will be with who is it next month? It is about Alaskan farmer to farmer exchange with Kristen Isfeld, Ken Sparta, and David Lee. So keep those things on your mind. And without any further ado, let's welcome Tom Kiffney. Tom is a graduate student at the University of Maine in the Brady Lab. And he is so gracious to be presenting with us today about diploid and triploid oyster research that he's doing. So take it away, Tom.
Thank you for that introduction. Let me just share my screen real quick. All right. So thank you. I'm a graduate student at the DMC. And today I'm going to be talking about some of my research that I've been doing over the last 2 years, looking at diploid and triploid oysters and how they perform in aquaculture settings, and a little bit about their physiology. And all of this has been done so far in the Damariscotta River estuary. So the theme of this talk is figuring out what those extra chromosomes are up to in these oysters. So just a little overview of what I'm going to be talking about today. So I want to give a little background about how we got into this research, as well as where we're doing this research. And then I want to talk a little bit about our growth trials. So just putting out these oysters in a couple locations and seeing how they perform. And then I want to talk a little bit about some physiology experiments we did. So we brought them into the lab and tried to figure out what was going on, what was going differently between the two ploidies. And then a little bit of extra side projects where we tested out triploid and diploids in some intertidal culture, which I'm pretty excited about. And so I just want to say too, if there's any questions during this talk, feel free to put them into the Q&A. And Sydney knows to interrupt me if there's something that I wasn't clear about, or you have more questions about, feel free to ask them during the talk as well. We don't need to wait till the end. So a little bit about where we do our research. So we are based down at the Darling Marine Center, which is UMaine's marine campus, which is based about halfway down the Damariscotta River estuary. So right where this arrow is pointing over here on the left. And it's a great place to do this work because up in the upper Damariscotta River is one of Maine's hubs of oyster farming. So it's been a pretty excellent place to study oysters, which has been really fun. And then if you look over here, this is actually our lease down at the Darling Center. So we have 3 tracks in this lease. So right here on this point, if you can see my cursor, this is where the lab is located. This is the pier where we dock our research vessel. But we have 3 tracks here. So this is our subtidal track right here that doesn't drain on low tide. So over here, this is a little picture of it. We just put out this new float, which we're pretty excited about. But this is where we have our long lines for surface or midwater culture. But then we have 2 separate tracks over here as well. We have one tucked up into Lowe's Cove over here that fully drains on low tide. And then we have another separate shallow subtidal tract right here in between these 2 piers. So this unique setup for our lease allows us to grow a whole bunch of different kinds of species on the farm. So currently we have sea scallops, eastern oysters, sugar kelp just got seeded last week by Adam St. Gelais. We've got some razor clams out there right now, and green sea urchins, as well as a few softshell clams. So that's a pretty exciting place to work. And so this farm is supported by the USDA and the Aquaculture Research Institute currently, but it doesn't just support the farm, it also helps support the hatchery at the Darling Marine Center. So seed is produced in here, as well as kelp. There's a kelp nursery, which allows us to then put those animals out onto the farm, test the grow out, host students to work on these projects, and then also do some environmental monitoring. So seeing how the temperature and any other environmental parameters that are interesting for oyster aquaculture we can measure. We can match that up to the grow out and hopefully inform aquaculture production around the state. And so I just want to briefly talk about some of the work that we have been doing and how this got us into triploids. So if you've been to some of our other talks, I gave one earlier, MAIC webinars on site selection. We have been doing a lot of work trying to understand how oysters interact with the environment to inform where we can site farms. And a lot of this work has been done to try to couple these responses to the environment to satellite data. So if we can look expansively across the state. And so part of this work has involved trying to create models of oyster growth. So figuring out how they really interact with food levels and temperature to help choose sites. And so I want to give a quick plug to one of the tools that has come out of this work, which is accessing high resolution satellite temperature data. So if you follow this link over here on the bottom or this QR code, it'll take you to this website, which allows you to click around and actually look at time series of satellite data at 100-meter resolution. So it's actually able to get into these estuaries and bays where we're doing aquaculture. And if you're a prospective farmer out there on the call and you have any questions about some of this data, my email is right down here at the bottom. Please reach out to me. I'm happy to help you walk through it, or if you have any questions about it. We're still developing this tool, so we'd love to hear from you about how we can make it better. But what happened when we were developing these models is we actually had a fair amount of growth data from some partner farms and from our farm as well. But we realized that we didn't actually do a great job all the time of recording whether we were looking at triploid or diploid oysters, which are the two types of oysters that are farmed in Maine. And so this guy is curious about how triploidy affects these changes that we were using to inform site selection. So it kind of spurred a parallel project to the site selection work, where we started to look at how triploids and diploids differ in Maine. So a little bit of background on triploids. There's two types of eastern oysters that are farmed in Maine. So they're the same species, but they differ in one main way, which is that triploids have 3 sets of their chromosomes, while diploids only have 2. And so triploids are often preferred by farmers around the world because of that extra set of chromosomes. That reduces their reproduction, so it allows them to grow faster. So the first reason that they like them, they grow faster, you can get a product to market quicker, which is usually desirable. And they also reproduce a whole lot less, which leads to a better meat condition on that half-shell market, especially while oysters are spawning during the summer, because no one really wants to eat that watery spawned oyster. However, this has not been a big issue in Maine, as our water has been typically pretty cold. But triploids are heavily used in the industry globally. So down in Virginia, about 80 to 90% of oyster production are triploids. And then over on the West Coast, about 50% back in 2018 were triploid oysters. And then even, internationally in France, about 100% of oyster culture relies on triploids. So they are used globally. And so that faster growth that is there are sort of 3 mechanisms that are proposed for that faster growth. And the first one is that they have a whole lot less reproduction, that they are more sterile than diploid oysters. So all of that energy that would have gone to creating gonads and eggs is now actually being able to put into the body of the oyster. And so if you think about it, you're not spending energy on something that is then broadcast out into the water column to create more oysters. There's a lot of leftover energy there. And the second is increased heterozygosity from having that extra set of chromosomes. So heterozygosity is when you have two different alleles for a particular trait on your two chromosomes. And this is just basically increased genetic diversity, which can give you an advantage in certain situations as those traits are expressed. And so this isn't actually a direct mechanism of faster growth, but it's basically increasing the potential to have an advantage. If you have three sets of your chromosomes, there's a higher chance of having some heterozygosity in there. And the last, which I think is very interesting, is that triploids often have larger cells. And I kind of think of this one as there's two ways that this can relate to a larger animal. If you have an animal that is built from the same cells, the cells are the building blocks. If some cells are larger, you'll end up with a larger animal. And there's also a chance of having a metabolic savings, which I'll talk about a little bit later, of those larger cells. And so those cells in triploids are a little bit larger because they have to hold a whole nother set of chromosomes inside that nucleus of the cell. And if the nucleus is larger, the ratio between the nucleus size and the overall cell size is conserved. So in general, triploids have about 50% larger cells. So these are kind of the three reasons that have been given for that faster growth of triploids. I also want to talk a little bit about how these triploids are produced in the hatchery, because there's actually been a series of methods that have been developed to get triploids into production. So the first method is called induced triploids, and these were the first developed actually at the Dowling Marine Center. So the first triploid oysters produced were produced here at the DMC in the 80s by Standish Allen and friends. This is the publication that's down here if anyone wants to look at it. So up here on the right, what happens when you mate a diploid with a diploid oyster? So the egg has two sets of the maternal chromosomes, and then it is fertilized with the sperm from the male. And then one of those maternal chromosomes is then pushed out of the cell, and your resulting animal is diploid. However, what they found is if you dump a whole bunch of toxic chemicals right on the oyster during that time, it can actually push that chromosome, which is being extruded, back into the cell, and you can end up with a triploid oyster, and the resulting oyster is triploids. However, this method had really low larval survival, which is not that surprising because there is a toxic treatment to it. And it did quickly become the main method of producing these triploid oysters. However, the chemicals were toxic and there was low larval survival, so there was a big push to find different ways to make these triploids. They tried different shock treatments, so whether it's temperature, pressure treatments, or different chemicals. I think they even tried using caffeine as one of the chemicals to actually induce triploidy. However, after Standish Allen left the DMC, went over to the West Coast, worked on triploid Pacific oysters, and then came back to the East Coast and developed the new method of creating triploids, which are mated triploids. So what they found—
Hey, Tom.
Oh, yes.
I'm so sorry. We did have a quick question related to, I think a clarification.
Yes.
Going back to the difference between triploid and diploids in so this question is asking, are you saying that the triploid diploid quote unquote spawny difference is less significant in cold water, Maine? Do diploids not taste as spawny in Maine oysters as compared to southern oysters? So I think this goes back to when you're talking about oysters being spawned out and tasting more watery as diploids.
Oh yeah, no, this is great. Thank you for clarifying. Yeah, so originally that has kind of been the common perception in Maine that the water here is a lot colder, so oysters are not putting as much energy into reproduction and spawning as much. Down in the Gulf of Mexico or Virginia, the Chesapeake, or some other places, these oysters can be spawning multiple times in the year. However, Maine is changing and people are noticing that oysters are spawning in Maine. So while reproduction might be a little bit less in these northern areas, it is not zero. And so one thing that we can look at is we don't have many natural oyster reefs around Maine as opposed to some other areas. So there is less reproduction of oysters in Maine, but it's definitely not non-existent, as scientists and groups around Maine have found that there are larval oysters out there, so they have to be coming from somewhere. So oysters are spawning in Maine. I hope that answered
Thanks
the
for that.
question.
Yeah, I
But
think
we can
so.
talk more
Yeah,
about that at the end as well.
cool.
So yeah, so going back to creating these natural triploids or these mated triploids, if you comb, while triploids do have much reduced reproduction, they are not completely sterile. And so what they did was they combed through a whole lot of triploid oysters until they were able to find some with enough eggs to do a spawn. They mated a triploid oyster with a diploid oyster, and then did that same chemical treatment that they used to create the first induced triploids to create a tetraploid oyster. So now this one has 4 sets of their chromosomes. And when you mate a tetraploid with a diploid, you can allow it to go through this natural process where they extrude 1 chromosome, because the egg of the tetraploid oyster will have 4 sets of chromosomes. The sperm from the diploid oyster will have 1. So there's 5, you extrude 1 and you end up, or there's 4, you extrude 1 and you end up with a triploid oyster. Sorry if I messed that one up. I'm still a little confused about this whole process too. But if you mate a triploid, or sorry, a tetraploid with a tetraploid, you can create more tetraploids. So you can maintain lines of these tetraploid oysters, which some hatcheries on the East Coast do. And they provide other hatcheries along the East Coast with these tetraploid broodstock to mate with diploids to create the triploids that are then provided to the industry. So if you're ever, going through the process of buying seed, sometimes you see the royalties fees that go back to the broodstock hatcheries for genetically selected lines. But there's also often an extra cost for producing these tetraploid lines as well, because it does take these groups money to provide these broodstock for free to hatcheries. So these mated natural triploids are now the current method used for U.S. oyster production. And it's really great because they have often high larval survival. So in the early tests, they were not statistically different from the diploid hatchery production, as well as that there's no toxic chemicals needed. So it is a much better method of producing these triploids. And I also just want to mention that it wasn't just Standish Allen working on this project as Shuming Guo as well down at VIMS. And so how well do these triploids perform in Maine? There is not a whole lot of information out in the literature. So when we got curious about seeing how these oysters performed, the first thing that we did was went to the literature and saw what was there. And so there's only been a few published studies actually looking at how well triploids and diploids perform. Some of that first work is from that first paper where they developed the triploids. And you can see here that there is sort of a minimal advantage from June to November in that second growing season with the triploids up here, about half a centimeter larger than the diploids. Then in 1992, also using induced triploids, a master's student redid some of this work and found that at 3 sites in the Damariscotta River, so out near Booth Bay, this blue site was at the DMC, and this red site was up near MAIC Sea Farm in the upper river. They found that there was no difference between triploid and diploids at all of these sites. We have the triploids in these triangles and dashed lines, and there was no significant difference in growth. So the first trial did find some, second trial didn't find much, and that has led to an idea in the literature that in the cold climate of Maine, triploids had minimal growth advantage because they are not spawning very much. There's a limited spawning season, which allows diploids to behave as sterile triploids. And so this is what all that information in the literature has indicated. And we wanted to test that again because the technology to produce these triploids, this was all using induced triploids. These new natural triploids are out there, but it hasn't been examined in the literature. And this is also just looking at the literature. The farmers out there have been doing their own trialing for many years, but we wanted to look at it in a systematic approach. So we wanted to test it ourselves. So this project started out with purchasing 2 millimeter seed from MAIC Sea Farm. So we got both ploidies. We wanted them to be the same, as close genetically as possible. So we ordered NEH by NEH triploid oysters and diploid oysters from MAIC Sea Farm. The diploids were a little bit older. They respond in mid-January, whereas those triploids respond at a later date in March. But we got the seed in June, and we put it out at 2 sites in the Damariscotta River. These are 2 of our interns that helped us work on the project, Eliza Grenchi and Lizzie Gilpatrick, which are absolutely integral to this project actually working. And so we put these oysters out in 2 environments. So we put them out at our farm at the DMC, and then also up at Pemmicote Oyster Company in the Upper River. And so another great part about this estuary as a test case for growing oysters and examining how they relate to the environment is this temperature gradient that we get along this river. So this is one of those satellite images looking at the Damariscotta River. And as you move up the river, you can see everywhere there's a natural constriction, right here at the Narrows or here at Glidden Ledge, the temperature increases. So in that same environment, we can get a warm site and a cooler site, as well as different food levels between the 2 sites. So another thing I wanted to mention is that we grew these in replicate floating ADPI bags. So no time was spent in oyster grows. We had nursery cage inserts, so very fine pet screen mesh that we slid into these bags to culture that early seed. And then we thinned them as they grew and increased the mesh size as well. So this is up at Pemmicote's nursery site in the Upper River. And then below that, this is our farm where we were growing our ploidies. And then in the winter, since this was a 2-year study, we took all the oysters back down to the DMC and overwintered them at our site before putting them out on their separate sites that next year. So in the summer, we measured them every 2 weeks. In the winter, we only measured them monthly. And so we wanted to see how they related to the environment. So over that entire study, we tracked temperatures and chlorophyll with sensors. And so this is the data from both those sites. In the blue, we have Pemmicote Oyster Company. So for both the chlorophyll here and the temperature on the top, the blue is the Pemmicote site and the black is the DMC. I'm just realizing now that I didn't label those. But so we have the 2022 growing season and the 2023 growing season. And you can see that at Pemmicote, waters were much warmer, reaching about 25 degrees over the summer, while the DMC sort of topped out at about 18 or 19 degrees Celsius over the summer. And you can see for food, we had 2 kind of different seasons. So food measured through chlorophyll was really high at Pemmicote Oyster Company in that 2022 growing season. It's a little bit of a fall bloom. It's fairly low over the winter and then picks up again in the next year. However, things were a little bit weird in this year, but we had all of that rain in the early summer, and we noticed that it kind of correlated with a dip in temperature when we normally get our peak temperatures. And that kind of correlated with the food at both the sites coming together a little bit. And that's because all of this rain could have affected the residency time in the Upper River, sort of washing out that warmer water and potentially washing out that food as well. So it was pretty interesting. We typically see seasons that look a lot more like 2022, but it's great to have an unusual season in this data set as well. But in general, Pemmicote Oyster Company was our warmer, higher food site, and the Darling Marine Center was our colder, lower food site. So how did things actually grow? So this is what we saw for growth, and I'm just going to walk through this this graph a little bit because it's a little bit confusing. So on the bottom here, we have shell height in millimeters. We have our 2022 season and our 2023 season. Blue here is Pemmicote, and the black is the DMC. These empty diamonds are our triploid oysters, and the closed circles are our diploids. And then on this top part here, we basically have percent advantage. So what percentage larger were the triploids than the diploids at both sites? So again, blue here is is Pemmicote Oyster Company, and black is the Damariscotta. And you can see right when we got the oysters, the diploids were older, so they were a little bit larger, but quickly the triploids took off at both sites and ended up being significantly larger in shell height, on an average of about 22% larger at both sites. And so I put this red line on here too, sort of just as an indicator for where market size is. So roughly a 3-inch oyster. And you can see that in that warmer, higher food site, the triploid oysters reach market size in just over a year, where the diploids took a little bit longer, but we're still reaching market size in the middle of that second growing season. Interestingly, the triploids at our colder site took off in that second season and also reached market size very in a similar timeline to the diploids at Pemmican Oyster Company, which is fascinating. The diploids were a little bit slower and kind of reached about an average of 50 millimeters in that second season. And so this difference in shell height was also seen in dry tissue weight. So this isn't the wet weight of the oysters. We basically shucked the oysters and dried them in an oven at 60 degrees Celsius for 2 days, sort of remove that effect of water weight to see how much physical tissue is there as well. And so this plot is set up in the same way. This is dry tissue weight in grams on the y-axis, our time on the x, and the same symbols and colors for Pemmican Oyster Company and diploid versus triploid. And this is the same process here. There was more variability over the season between the two ploidies and percent advantage of tissue weight. However, on average, over both sites, there was about a 50% advantage in tissue weight in triploids. The triploids ended up being absolutely massive by that end of the second season. This is about a 10-centimeter oyster in September with about 4 grams of dry weight. So one thing that we wanted to do as well is just to see if these oysters are shaped any different. So these are the means right here. So on the last sampling day, we took the oysters in and on about 120 oysters for each of those sites and ploidies, we took different dimensions. So not just how long the oyster was, but how wide it was and how deep it was as well. So this first plot here on labeled A, this is basically some box plots looking at those differences. And so what we're looking at here for a box plot, this dark line is the median value, so the middle value. And then the square is the top, about 50, represents 50% of the data. And then these whiskers are the extra 25%. So it gives you a better view than what just looking at the mean and the standard error is. So this is our DMC diploids, our smallest oysters with a median value about 60 millimeters. The DMC triploids and the Pemmican diploids were not statistically significant from each other at the last measurement day. They had practically the same shell heights, whereas the POC triploids were much larger, closer to that 100-millimeter oyster. So this fan ratio is basically the height of the oyster divided by the width, which basically means that the higher value is basically a skinnier oyster. So we can see that in both sites, the diploids were closer to 1. 1 would be an oyster that is as wide as it is tall, so a perfectly round oyster. So in both cases, the triploids were skinnier oysters significantly. As for cup ratio, which is basically the ratio between how long the oyster is and how deep it is in this cup, so basically how nice of a deep cupped oyster do you have, we saw that the DMC the only statistical difference was between the Pemmican diploids and the Pemmican triploids. Higher value is a deeper cup, and the diploids were slightly deeper cupped than the triploids at that site. So takeaways from the growth trial. So mated triploids grew much faster than diploids at both of our sites, which is contrary to kind of that information that is out there in the literature right now. Market size in colder areas that only reach about 18 degrees can be reached in that second growing season if you are growing triploids. And there's some indication in a future webinar, maybe we are doing some regressions to actually pull out the relative effects of food and temperature on ploidies to make some better recommendations about site selection. But however, right now that data kind of indicates that triploids have a higher advantage in warmer waters that have more food. But that will be the subject of maybe a future webinar. And so also I want to note that diploids have slightly better half shell characteristics. So maybe as they are growing faster, since they are growing slower, they're more susceptible to some of that chipping that can come with the movement of the bags, creating a better shaped oyster.
Hey, Tom, we have a
Yes,
couple more questions about your
absolutely.
growth trials.
Yeah.
One of them, we have a question, were the stocking densities maintained equally in all treatments?
Yes, they were maintained equally in all treatments. So we initially stocked them at about 2,000 oysters in those, about 1,000 oysters in those nursery insets at both sites. And then when we thinned them, I think about to 804 millimeter mesh, and then down to 150 in that second growing season. They were always thinned at the same time at both sites to make it consistent.
Cool, thanks. And then we also had another question.
Yeah.
This person was wondering why 3N shell height would have decreased at times, and in one of the 2N groups too, like in the fall of both years and in the spring of one. Maybe there was some disease causing mortality that affected the largest oysters.
Yes.
That was one hypothesis.
What more likely is happening are these are probably our thinning events. So there is some natural variability around here because we also, we're measuring a random sample of these oysters. We have 3 bags per ploidy per site, so 12 bags overall. We measure 40 oysters from each bag, but randomly selected. So there is a little bit of variability in there. But then what also happens is when we thin the bags, sometimes it changes the overall mean. So that's what could have happened here. We also could have just maybe grabbed the smaller oysters by accident when we were doing the measuring. So there is a little bit of variability in here. But overall, it's sort of an artifact of the measurements.
Gotcha. Yeah, and then we had a couple more questions related to mortality. People are just super excited about this, which is sweet.
Awesome. Okay.
How does mortality compare between diploid and triploid at both sites? Did you measure that at all?
That's a really great question, and I am wishing that we did measure it. I'm going to show some data from the intertidal experiment that we did with Dana Morse, where we did actually track mortality over a season. So we will get to that later. But unfortunately, we did not track mortality during this experiment. I can say from visually, mortality was very high in both ploidies. We did not lose a whole lot of oysters over this trial, just from looking at, empty shells that we did see. We did see some mortality, but nothing that would have raised any concern.
Sweet. Okay, we'll leave it there for now and come back to more later.
All right. Also, please, my email will be at the end of this. Please reach out to me if you have any other questions or would like to chat. Okay, so moving on. So now I want to talk a little bit about some of the physiological studies that we did to kind of look at, what could be playing into this advantage for triploids. And so one that I was mostly excited about was the idea of these different cells. And so I was kind of curious to see if there could be a metabolic saving of having these larger cells in triploids. So a lot of the work that we do into maintaining our bodies is actually revolves around maintaining gradients in our cells. And it can be more efficient to maintain gradients in cells that have lower surface area to volume ratios. So the larger cells in triploids may be more efficient, and that means that less energy may be used on maintenance, meaning that more energy could be used for growth. And so the way that we went about measuring this is putting oysters into tanks and starving them. So we want to actually get at measurements of maintenance needs. So we need to remove any other metabolic need that's going on. So we spend energy on feeding, we spend energy on growing, we spend energy on a whole lot on reproduction. So the only way to get rid of those components is to put them in an environment where they are no longer feeding, they are using up the reserves that they could be using for growth, and they are no longer putting energy into reproduction. The idea is as they starve, we can measure how much oxygen they are consuming and see if there is a difference between the ploidies. But first we wanted to see, are the cells actually even larger in triploids? So we had Tobin Strevey, a student at Maine, who's been in the Semester by the Sea program at the Darling Marine Center, and also a Norumbega crew member, go through all of these histology slides that we took of triploid and diploid oysters and measured the surface area of hemocytes, these cells here, that we found that we could see a nucleus, that's that little dark purple spot. So basically measuring the surface area of all of these cells from about 12 oysters per ploidy, and he measured 30 cells per oyster. And so we're seeing over here on the right, this graph is basically that cell surface area. You would draw a little box in image J around the cell and calculate the area. Well, we can see that in the triploids, they do have larger cell surface areas, which is quite exciting. And on average, triploids had about 37% larger volume of these hemocytes. And so I say volume in quotation marks because that's basically just assuming the cell is a sphere, which they're not, taking that diameter from the surface area, assuming that we cut right through the middle of the cell, which again is a reach, and then calculating a volume of a sphere. So, but we did get closer to that theoretical 50% larger volume that is out there in the literature. So that was exciting. So yes, triploids that we saw did have larger cells than the diploids. So then we were like, great, let's do our starvation experiment. So what we did was we brought our animals in from the overwintering. So this started in November. We took overflow animals from the Pemaquid Oyster Company trials for triploids and diploids. We put them in these 8 replicate tanks that we have in the flowing seawater lab, and we slowly ramped them up to 24 degrees, and then we acclimated them there for 2 weeks. And so during this time, we fed them 2% of their body weight in algae. You can see these fun little IV bags we have here dripping down into the tanks. And then after that temperature ramp and acclimatization, we cut off food and we measured respiration and tissue weight for 10 weeks. And so this is how we measured that oxygen consumption in the oysters. Each one of these chambers here has a little oyster in it. We flush the chambers with fresh water, or not fresh water, fresh salt water, and then cut off the exchange of water and measure how fast oxygen declines. And we did that regularly while we were measuring how fast they were starving. I'm running out of time, so I'm gonna blaze through this part a little bit. But this just shows what we were looking at. So this is the experiment day. We had about 20 days of temperature ramp and acclimatization. Then we cut off food, and then we watched them starve. We did see that during this temperature ramp and feeding period, we actually did lose a significant amount of weight in both triploids and diploids. And this is likely because we were kind of bringing them out of that fall quiescent period and likely not giving them enough food, given that we are ramping them up to 24 degrees, even though we did it slowly. But overall, we saw that both animals lost weight and that the triploids were always larger than the diploids here. When we actually standardized by how much shell each oyster had, we start to see that triploids starve slower. So on the y-axis here, we have condition index, which is basically just the meat weight divided by the shell weight times 100. So it's a way of standardizing for different sizes, assuming that they're not putting on any shell while they're starving. We checked this as well, and shell height was stable across the entire experiment. So we do see that they started out with similar condition indexes, but right off the bat, they started to diverge. And so triploids had higher condition indexes through that entire experiment. And this can be seen as this experiment progresses. We start to see higher mortality in the diploids than the triploids here. And this is just cumulative mortality, the number that has died over the course of the experiment. And we don't really see this uptick in diploid mortality until around day 40. So great, we saw slower starvation in our triploid oysters, and we thought, awesome, this is probably related to a lower metabolic need. However, we saw no difference in the statistical difference in the oxygen consumption data. Here we have how much oxygen consumption standardized to a 1 gram oyster that these oysters were using. And we only started this after this temperature ramp period. It would have been interesting to have a pre-measurement, but we didn't do that. But we saw that at no point was there a difference between the triploids and the diploids. So physiological takeaways, that triploid cells were larger. The triploids appeared to starve at a slower rate, but that slower rate may not be due to lower maintenance needs. We measured about 18 oysters per ploidy in each of these trials. So it's possible that we did not have the numbers to actually find that small difference that might be between them, but there could be other factors that lead to this as well, such as even just behavior, where the triploids opening up more trying to feed and using, or sorry, the diploids opening up more, trying to feed and maybe using more energy. But we'll hopefully be looking into that in the next few weeks. And now I'm going to move on to talking about the intertidal zone for maybe the last 10 minutes or so. I just want to pause to see, Sydney, are there any questions about me blazing through that right now?
We did have a couple questions.
Yeah.
I think just related to the differences between diploid and triploid. We had someone was wondering about if there were differences in disease resistance and any differences in shell densities. Those are kind of big questions. So.
Yeah, so we didn't look at any of those specifically in our work. There has been I think some work out there in the literature saying that they sort of respond to diseases the same, and there's been some very recent work saying that no, they actually respond quite differently. But I don't have those answers right off the top of my head, unfortunately. As opposed to shell density, that is definitely something that I've heard from farmers. When you have these oysters growing really, really quickly, especially these triploids, sometimes the report is that the shells are more brittle and crumble a little bit more when you shuck them. What I noticed when I was, shucking all these oysters for this project, I didn't see a huge difference between the two, but it'd be very interesting to take a closer look at that in the future.
Great, thanks, Tom.
Okay, so now on to the intertidal zone. And so this is work that we have been doing with Dana Morse of Sea Grant, looking at intertidal culture in Maine. And over this last year, we sort of put some of these triploid and diploids that we had on our farm into this intertidal tumble culture, which has been pretty exciting. And we just got this data, so it's not as robust as what we were looking at before, but equally exciting. And so this has been a topic that's been raised in a couple of MAIC webinars. I think Amanda Mosier mentioned this from Mexico, and Kyle Pepperman mentioned intertidal farming and how prevalent it is in their trips out to Washington. But intertidal farming does exist in Maine. So this is data from MAIC's R&D&E surveys. So in 2022, there was no one working with intertidal culture systems. That jumped up to eight farmers in 2016 and has held steady at around 12. These aren't exact numbers. These are from me squinting at the plots. So I might ask Chris for that actual data, Chris and Annie. But there's two methods of intertidal farming. One that's used a lot is called rack and bag. So basically having these structures that are fixed in the intertidal zone and then placing your oysters on top of that. Which seems pretty great because you can use your ATV and get out to your farm. And the other method, which basically is called a bunch of different names: the adjustable longline system, intertidal longlines, tumble culture, also called flippy bags by a lot of people. So basically, this involves having a fixed longline in the intertidal zone, and then bags or tubes that are attached at one side with a float. So when the tide comes in, that float flips the bag, the oysters tumble, and you get kind of a 2-for-1. So when the tide is out, these oysters are desiccating, killing off some of that biofouling. And when the tide comes in, you get a natural tumbling action. So that kind of leads to the pros and cons of intertidal oyster farming. Some of the pros are you have to spend a lot less time going out to your farm, flipping your oyster cages, allowing your oysters to dry, and a whole lot less time taking those oysters out of the bags and tumbling them yourselves. Possibly it could lead to a better shaped oyster if it is tumbling, every 6 hours with the tide. This could be a great way to actually form that shape, chip the growing edge of the shell, so you get sort of that stronger shell, those deeper cups, and those wider oysters. Also, you might not need a boat to harvest on low tide if you can walk out to your site, which would be a huge cost saving. There are some possible cons with intertidal oyster growing. Possibly all that chipping could lead to slower growth. You also can only access your farm on those low tides, which, around this time of year, if they line up with, the nighttime at 4 o'clock, you can be out of luck trying to get to your site on low tides. And also in Maine, we have cold winters, so we still need a deeper lease to overwinter those oysters. So you'll have to bring everything off the farm over to another lease to overwinter them. So in recent work that Dana has been leading down at the Dowling Marine Center, there's been a series of small-scale projects underway looking at different types of gear, as well as different density effects in these intertidal systems, mostly looking at ADPI bags on different orientations attached to these long lines, SEPA bags, and Zapco tubes, which are two forms or two growing containers that are actually designed to grow on these long lines. These are some examples from some partnerships with MAIC Sea Farm up at their intertidal site in the upper Damariscotta River using rebar staples, and over at Skelly Sea Products using, we have these ADPI bags in different orientations, the SEPA bags over here as well. And then this is just an example of really, really high stocked Zapco tubes with all of these strange deformities that we see on these oysters growing in weird shapes, and then some really beautiful low density SEPA oysters. So some of this recent work has kind of highlighted that trade-off in a well-shaped oyster and the growth. So this is a combination of work from 2021 and 2022, basically looking on the x-axis here, we have change in that cup ratio. So how deep is that oyster? As you move to the right, we're getting deeper cupped oysters. And as we move up, we're getting a faster growing oyster. So in the upper Damariscotta River above Glidden Ledge from 2021 to 2022, there was no trade-off. This line between deeper cupped and growth rate was perfectly flat. Whereas down at the Dowling Marine Center, we actually saw a trade-off at our site, where when we got deeper cupped oysters, they had a much lower growth rate. So we wanted to look into this a little bit more. One of the two differences between our upper river sites and our lower river sites was whether they were a shallow subtidal site. So these oysters were being exposed or were being submerged in the water more than we were getting at our DMC site, which was more of a true intertidal site. So we had all these oysters left over from our polyploidy work. So we were wondering, can oysters be grown for 1 year in surface culture, which we had been doing for the study, and then put them in that second year into Zapco tubes in a shallow subtidal site and an intertidal site, and see if they can be reshaped into a better shaped oyster. And these are the Zapco tubes that we've been using, which are about 36 inches long, and then they're a round tube and about 1 foot in diameter. And they kind of flip around this line. There are floats in the caps of these. So this is kind of where we had our site designed. I'm going to probably speed through this a little bit because it looks like I only have 10 minutes left. But this is, again, our lease site. We have our intertidal site over here. We have our shallow subtidal site right here. And in the graphs that follow this, just going to be called our subtidal site because our floating lease over here is going to be our control site. So again, these oysters were grown for 1 year in the floating ADPI bags and then moved to the intertidal and control site. We put depth sensors out at both these sites. So this is the water depth in orange at the shallow subtidal site and in blue at our intertidal site. We can then calculate the percentage of time that these oysters are actually exposed to air. So in the intertidal site tucked up in Lowe's Cove, they are reaching about 30% of the 30% of the day exposed on these large tides. So that's about 3 hours on each low tide, where the shallow subtidal site was only getting a couple hours on each tide. And there are some cases where the intertidal site was being exposed and the subtidal site was not seeing much exposure. And when we broke down the study, we actually had some help from students from the last aquaculture and shared waters class. They came down, helped us break down the experiment and measure all these oysters, which was a really fun way to have some help and keep Dana and I from going crazy measuring hundreds and hundreds of oysters. So is there a difference between these sites and different ploidies? So we have here in these graphs, we have what we saw before. We have shell height, we have our cup ratio and our fan ratio. In the gray for both of these is the initial site. So these are the measurements right when we put them out. And then in the red and green are different ploidies. And these are the measurements that we took when we took them in October. And so from shell height, we saw that in both cases, there was an increase in shell height from our initial measurements. But there was no difference between the controls. These were the floating bags out on our subtidal lease, and then our intertidal and subtidal. So they were all not statistically different. So there was no reduction in growth with some of that tumbling that we saw. And that could be a little bit clearer when we look at these cup ratios. So here we have our larger values, our deeper cupped oysters. And we saw for our diploids an increase in our cup ratio from our initial values for all of our sites. But we didn't see a difference between our sites. And that's the same for our triploid oysters as well, where both of them became deeper cupped oysters, but there was no difference between our floating farm, our intertidal, or our shallow subtidal farm. And then fairly similar, but for fan ratios, we saw that actually there was no significant increase in our diploids from our initial values compared to our controls and our intertidal and subtidal. The only difference that we saw here was that our intertidal farm, there was a significant increase in fan ratio. So larger value here means a rounder oyster. We're getting closer to that one value. This was the only significant change, meaning that these oysters increased from that initial value. So no reduction of growth compared to intertidal systems. Both ploidies gained deeper cups, but only intertidal triploids increased their fan ratio over initial values, but not more than the control or the shallow subtidal plot. And these are just some pictures that we have of the different ploidies from breaking down the experiment, just to show that these are not to scale, so don't take the size into account. But just that you do lose some of this fluting that we see in the diploids. These are our control, our floating bag, triploids, our floating bag, diploids, and then our intertidal systems. And so one thing that could be leading to this difference that we don't see here is that we are using just floating bags. We're not using oyster grows, which do reduce some of that motion when waves and wind are causing some turbulence at your farm. But we do see much smoother oysters over here, as well as much rounder ones that we're seeing in these intertidal diploids, which is pretty interesting. And to get a little bit to this mortality, we did track mortality at all of these sites over that summer. And diploids appeared to have higher mortality than the triploids across treatments. So we did see roughly 12% mortality for our diploids, and then about only a 5% mortality for our triploids at all. And there was no difference between these sites, which is pretty interesting. But overall, these values are quite low for mortality, but extremely low for our triploids. I just want to thank everyone who made this work possible, our partner farms, Maine Oyster Company, MAIC Farm for helping out, and Skelly Sea Products, as well as our funding through the USDA, the School of Marine Science, and the Aquaculture Research Institute. Any questions? I also want to thank Jess Capista for making me these awesome title and end slides.
Thanks so much, Tom. We have two more questions related to the last section there. So the first one is, did you notice a difference in predation and mortality? You talked a little bit mortality, but predation between the subtidal and intertidal areas.
Yeah, so in those systems, they were all contained. So we were not bottom planting anything. They were all held within those Zapco tubes. So mortality was quite low. From predation, at least. There was no green crabs getting inside our bags.
Oh, all right, thanks. Any difference in the ability to deal with big changes in salinity?
Yeah, so if there has been a lot of work out of the Chesapeake and the Gulf of Mexico to look at this. It is harder for the triploids to sort of change that, maintain the volume of their cell, possibly, when they're faced with these big swings of salinity. However, in the Damariscotta River, even on a really rainy season, our salinity was still quite high, typically above 25 PSU. So we weren't seeing that in our data as well. But if you want to talk more about that, I'm happy to look at that literature as we keep working through this data, and we can have that conversation.
All right. And one last question about any differences in taste.
In taste. I don't know. There's been, I'm going to be honest, as I keep doing this, I have eaten fewer and fewer oysters, the amount of time that I spend on the farm. So I haven't tasted them. There has been some indication in the literature that intertidal oysters actually are sweeter. They build up those glycogen stores to help continuously repair those shells. But I would love to have a blind taste test and see if we can find differences between intertidal and subtidal oysters, as well as between ploidies. I've never heard of anyone tasting them differently, except during the spawning season.
Neat. Well, that wraps our questions. Maya, are you giving a little wrap up?
Yeah, sure. Thanks for asking all those questions. And Tom, great job. Thank you so much for presenting. That was really interesting. People seem to really enjoy it. For anybody who joined late, we are recording this webinar and we will post it and send it to everybody. So that should be up sometime next week. And just to reiterate the reminders, oh wait, I think we did have one more question come in. Sydney, do you want to jump in and read that first, and then I'll say the reminders?
Yes, this one is a little bit more of a paragraph, but in the first part, the growth curves between the diploids and triploids seemed more divergent in the first summer and are more parallel afterwards. Why do you think the diploid seed had a slower start? Do you think there's a lot of an effect, or because of the diploids were older and spawned in January versus the a little bit younger triploids?
Yeah, that's definitely a possibility. The growth rates in oysters slow as they sort of reach those maximum sizes. So as animals as oysters get bigger, those growth rates are decreasing. And so since triploid oysters get to a larger size, about 20% longer than their diploids, potentially they're reaching that maximum size a little bit later, but it also could be related to the age as well. And we're doing some more work now to see if we can also pinpoint if it's them responding to the different environments as well, potentially being able to take in more food or respond to higher temperatures. So hopefully we'll have more answers on that question if Chris and Annie let me do another webinar.
Stay tuned for next year. Yeah.
Yeah, so that's great launching point. Thanks for answering that question, Tom. So next year, January 19th, again, that's an Alaskan farmer to farmer exchange webinar with Kristen Isfeld and Ken Sparta and David Leith. NACE happening January 10th through 12th in Providence, Rhode Island. Get your registration now. Also sign up for the field trips and workshops. They're super exciting. And register for the early bird hotel discount before the 22nd. And finally, if you are interested in specific webinar topics, feel free to email one of the MAIC team members or the MAIC Gmail account, which is available from the Contact Us page on our website. And we would be happy to kind of adapt our webinars to the interests of our community. So that about does it. Happy 2023. Yeah, that's this year. Happy 2023, folks, and see you next year, I guess.